Hi. I first would like to thank the organizers for the opportunity to speak here. My talk will be on the computer-aided design of liposomal drug in silico prediction and experimental validation of drug candidates. Our computations are based on chemoinformatic methods developed in our lab for identifying molecules as drug candidates for a particular biological target. In this study, we used these methods to identify molecules as candidates for liposomal delivery. And the motivation was that drug delivery system may improve drug toxicity, pharmacokinetic, and biodistribution, but the testing of drug suitability to a liposomal drug is time and labor consuming, and by computational approach, we can save time and labor. We chose liposomes as our delivery system because of their advantages, and most importantly, because we had enough data to create reliable models. The approach presented here may serve for candidate identification out of large molecular libraries and for the designing of chemical modification for a specific drug to fit a liposomal formulation. In general, there are two main methods for drug loading into liposomes. The first one is passive loading, in which a drug and lipids are co-dispersed in an aqueous medium, and drug is loaded into liposomes while liposomes are being formed. This method usually yields a low loading efficiency and low drug concentration in liposomes. The second method is active or remote loading, and in this method, drug is loaded into prepared liposomes by pH gradient. This method is suitable for amphipathic weak acids or bases, and if suitable, may result in high and stable loading. In this study, we concentrated on remote loading and built models that will be able to identify suitable molecules suitable for this delivery system. This is an illustration for remote loading of amphipathic weak acid by transmembrane calcium acetate gradient. Liposomes containing calcium acetate in their intraliposomal volume are prepared, and a drug in its unionized form crosses the membrane. There it forms insoluble salt with calcium, and the acetate which goes to the outside of the liposomes generates the gradient. This method is therefore suitable for amphipathic weak acid or bases, which we define by having log D in the range of -2.5 to 2, and pKa value of below 11 for weak bases and above 3 for weak acids. So in order to build the models, we collected data related to remote loading of liposomes, and we defined the target property as the highest initial drug-to-lipid ratio that will enable high loading efficiency. As we were interested in developing a liposomal drug, we did not need only high loading efficiency, but also high drug-to-lipid ratio in the liposomes that will enable administration of a therapeutic dose. That ratio we defined was 0.3, and that way all molecules that had low loading efficiency at this ratio were regarded as negatives, and molecules having high loading efficiency at this ratio were regarded as positive. The final data set consisted of 46 drugs studied in 143 experiments, most of them, 112, were negative drugs, and 31 experiments were classified as positive. Each instance in the data set was described by properties, also called descriptors. We had two types of descriptors: drug delivery system descriptors, which are descriptors that related to the formulation composition and the preparation process. And we had structural descriptors, which are descriptors related to the structure of the molecules. And these are computed by suitable software that's used for other QSPR studies. This is a general illustration of QSPR studies. We have the chemical structures that are transformed to descriptors, and with the noun property, they're translated to modeling by computational tools. Then the models that are generated are used for screening of a large molecular database, and we can see in the illustration the separation between actives and non-actives. We use mainly two classification methods. The first one is iterative stochastic elimination, which is an algorithm that is used to solve diverse and extremely complex problems. In our case, we had instances that each described by 194 descriptors. So the first stage was to find the optimal range for each descriptors, and then to create filters. And each filter is produced by combining gradients of a few filters. So in order to find the optimal filters, the optimization consists of stochastic phase in which variables that contribute to the worst solutions are iteratively eliminated to a point that the search space size can be calculated exhaustively to find the best filters. And then when we have the best filters, we can use them to screen large molecular libraries, and a molecule that passes more filters gets higher index. And this is an example of one of many, many filters that we received. The second method is k-nearest neighbors, which is a very known method, data mining method. In this case, we create a chemical space by the descriptors, and the unknown molecule property is predicted from its neighbors in the descriptor space. I'll skip this slide. So we had the models that we generated, and we used them to screen the large library. We had the comprehensive medicinal chemistry library, which is composed of more than 8,000 molecules. And you can see this figure, which is a result of the virtual screening. Each dot in the figure represents the score of a molecule by both models. We can see that most of the molecules in this area were predicted to be negative by both models. And only a few, 2.3%, were regarded as positive by both models. And then we had to select candidates for experimental testing. In the first stage, we were left with 197 positive molecules by the virtual screening. And, just a moment. And now, in order to select the candidates, we implemented other considerations which relate to liposomal development, which are the availability of enough drug for the testing, the indication for which the drug is intended and that it will have any therapeutic advantage to give by liposomal delivery, the dose that should not exceed 200 milligrams, and of course that it will fit the requirements of being of hepatic weak acid or base. We finally selected 3 molecules, 2 positives, pravastatin and mupirocin, and 1 negative molecule. We can see that all molecules fulfill the requirements of being of hepatic weak acid or base, but the models distinguish piroxicam as a negative molecule. And this is where the molecules are found in the virtual screening figure. We can see that pravastatin and piroxicam are located here in the positive side, and piroxicam is in the negative side. The results of the experimental testing confirmed our predictions, and we can see that pravastatin and mupirocin were highly loaded at all drug-to-lipid ratio that were tested, and piroxicam was low loaded at the ratio that were tested. We additionally validated the models with 10 additional molecules that were published in the literature that were remotely loaded to liposomes and were published after we established our models. So we can see we had 10 molecules, out of which only four were suitable for remote loading, meaning that they were amphihepatic weak acid or bases. The models misclassified 1 positive molecule, and KNN also misclassified 1 negative molecule. But the overall accuracy of the models found by the external validation by our testing and by the literature was high. So as a result of this modeling approach, we developed nanomupirocin, which is a pegylated nanoliposome that was remotely loading with the antibiotic mupirocin. Mupirocin was found to be positive by the models, and it presented a special advantage for liposomal delivery. Mupirocin is an antibiotic with a unique mode of action, not shared by any antibiotic in the market, and it is active against resistant bacteria, but its use is limited to topical application due to its PK properties. We thought that liposomes, nanoliposomes, may enable the administration, the systemic application of mupirocin because it would protect it in the circulation and passively target it to the infected tissue by the EPR effect. So indeed, in a PK study that we did, we saw in this figure, we saw improved pharmacokinetics of nanomupirocin versus free mupirocin. The AUC ratio is 100 to 1. And you can see that even 24 hours after administration, we still have enough concentration, which is higher than the MIC of important bacteria that are presented here. We also showed efficacy of nanomupirocin after IV administration in a mice necrotizing facilities model. In this model, we inject group A streptococcus subcutaneously to the back of the mice. And in the first short movie, we can see the sick mice, the untreated mice, they barely move, and their hair is rough. They have big wounds on their back. This is 24 hours after the administration of the bacteria. And in the nanomupirocin group, they are not sick. They show known symptoms of the disease. If we look on the survival rate, then all the animals in the nanomupirocin group survived the study, while only 1 mouse out of 6 survived day 3 after administration of the bacteria. So we can say that nanomupirocin enabled the parenteral activity of mupirocin, and we may say that it's a new parenteral antibiotic. So I prepared this slide just because I know that experimental scientists are very suspicious towards computational approaches. And I want to show the capabilities of this approach for screening and as a tool in liposomal development. The 4 molecules presented here were remotely loaded to liposomes, and only 1 is considered to be a good candidate, meaning that it was highly loaded at drug-to-lipid ratio of 0.3. If you can just try to find the correct one, I also added a pKa and log D value, which also takes time to compute. This is the correct one. So if we think of the time that it took you as professionals to screen 4 molecules, and considering that a computer can do that in less than 5 minutes, you can see the advantage of this tool. I would like to thank all the people that were part of this project, members of Professor Goldblum's lab and Professor Barnard's lab, Alex Stropsha, that helped in the computational part, and Professor Alon Moses and Dr. Ayelet Gallego from Hadassah hospitals that did all the in vivo study with nanomupirocin. And thank you for listening.
Thank you very much for your really nice presentation, very helpful, I think. We can save a lot of time just using your tools. Is it possible to use your tools? So if you come with
you can contact
an idea?
us. Yes, you can contact us and we can do it for you.
Okay, any question from the audience?
Okay.
Thank you again, Dr. Cern.